Welcome to the Explainer. Today, we're zooming past the basic mechanics of CRISPR to unpack something much heavier: its profound ethical responsibilities. We all know how the molecular scissors work by now, but as this technology aggressively shifts from the lab into the clinic, the conversation has got to shift too. We need to move away from simply asking what CRISPR can do and start asking what it should do.
Are we ready to edit the human germline? Honestly, this is the jarring central dilemma of our time. It's not science fiction anymore, you know? The capability to rewrite human history is literally in our hands right now. And that is forcing us to grapple with unprecedented moral, societal, and ecological questions, all before the science completely outpaces our ability to regulate it.
So, here is our roadmap for today. We're going to hit the He-Jiankui affair, look at precision and off-target risks, dive into the 6 major ethical concerns, the designer-baby debate, ecological risks of gene drives, and finally, what the future of gene editing looks like. Let's jump right in.
Starting with Section 1, the He-Jiankui affair, a case study in ethics violated.
Now, to really grasp why this scandal blew up the way it did, we have to distinguish between somatic and germline editing. Somatic editing only affects the patient being treated. Germline editing, on the other hand, permanently alters the DNA of embryos, sperm, or eggs. And the kicker here is that these changes don't just affect one individual, they're passed down to all future descendants. It completely bypasses the entire concept of future consent.
So, back in 2018, Chinese biophysicist He Jiankui actually did it. He edited human embryos targeting the CCR5 gene to try and confer HIV resistance, which resulted in the birth of twin girls Lulu and Nana. And when he announced this at the Hong Kong summit, the global scientific community didn't celebrate. They were absolutely horrified. It was medically unjustifiable, ethically unacceptable, and it ultimately led to his conviction and imprisonment for illegal medical practice.
All right, moving on to Section 2, precision and off-target risks. Basically, just how precise is CRISPR in the real world?
Well, it's powerful, but it isn't perfect. A massive risk in this space is unintended off-target cuts. Think of it kind of like a typo in a search engine. If the guide RNA tolerates a slight mismatch, those molecular scissors end up cutting the wrong DNA. And this risk goes way up if the CRISPR machinery is left lingering inside the cell for too long, basically giving it more time to make mistakes.
But scientists aren't just crossing their fingers and hoping for the best. They use a really robust toolkit of advanced screening methods to diligently map out those dangerous cuts genome-wide. Methods like GuideSeq can actually flag an error down to a fraction of a percent in living cells, while others work in a test tube to remove biological bias. The whole point is verifying that our edits are exactly as precise as we intend them to be.
And fortunately, we are actively engineering these risks out of the system. By tweaking the Cas9 protein itself to weaken nonspecific DNA contacts, variants like ESP-CAS9 are seeing a tenfold reduction in off-targets. Then you have things like Hi-Fi Cas9, which maintains high on-target activity even in those really stubborn, hard-to-edit primary cells. These engineered proteins are absolutely vital for safety in clinical settings.
Which brings us to Section 3, the 6 ethical concerns shaping our moral landscape.
Pretty much every application of CRISPR is going to bump right up against at least one of these 6 major societal anxieties. You've got the heritable germline editing we just talked about, right alongside equity and access, consent and autonomy, the very blurry line between enhancement and therapy, ecological risks, and of course, dual use and biosecurity threats.
Let's make that equity and access concern super tangible for a second. $2.2 million. That is the per-patient cost of Casgevy, the approved CRISPR therapy for sickle cell disease. Now, considering sickle cell disease disproportionately affects regions like sub-Saharan Africa and India, a price tag like that is alarming. We're currently risking a 2-tier world where genomic medicine is this incredible miracle, but it's reserved exclusively for wealthy nations. It demands some serious conversations about global pricing.
That disparity flows right into Section 4, the designer-baby debate. Where is the line between enhancement and therapy?
Reporting strictly on the current consensus, this debate is deeply, deeply polarized. On one hand, you have utilitarian arguments asking, well, if we can safely prevent severe genetic diseases before birth and literally reduce human suffering across generations, isn't it our moral obligation to do so? But the counter-arguments are massive. Critics point out that the technology violates the autonomy of future generations and opens a terrifying slippery slope to eugenics, where editing for disease morphs into editing for intelligence or height, risking a dangerous homogenization of human genetic diversity. All right, zooming out from human genetics, let's hit Section 5, Gene Drives and Ecosystems, the Ecological Risks.
These broader environmental anxieties are perfectly illustrated by gene drives. A gene drive is a CRISPR-based system that literally overrides normal biological inheritance. Instead of the usual 50-50 chance of passing on a gene, this system actively copies itself onto the homologous chromosome in every single generation, forcefully pushing a specific mutation rapidly through an entire wild population.
We're talking about a near 100% inheritance rate. That is an incredibly powerful tool, often proposed for things like wiping out malaria-carrying mosquitoes. But releasing a gene drive organism carries an irreversible ecological risk. Once it's out in the wild, you can't really put the genie back in the bottle. It could permanently alter ecosystems or inadvertently drive whole species to extinction. And right now, international governance frameworks are definitely lagging behind the science.
So, how do we prevent a runaway ecological disaster? Well, biotechnology utilizes anti-CRISPR proteins. And a pretty cool twist, these are actually born from an ancient evolutionary arms race between bacteria and the viruses that infect them. Proteins like ACR-IIA4 act as molecular safety switches. They physically mimic DNA, blocking the Cas9 scissors from ever binding and cutting. Having this off switch is arguably just as crucial as having the editing tool itself.
Finally, Section 6, the future of gene editing. What actually lies ahead?
The next decade is looking absolutely wild. We are moving toward much smaller proteins, like Cas5, that are way easier to deliver into the body. We're seeing PAM-less variants that dramatically expand the areas of DNA we can actually reach. Throw in AI-guided design and epigenome medicine, which can change how genes behave without even altering the underlying sequence, and it's clear CRISPR cementing itself as an unstoppable, evolving force in modern medicine.
So, as we wrap up this explainer, I want to leave you with a final thought. We've seen the mechanics, we understand the risks, and the potential is obviously boundless. The power to literally rewrite the code of life is firmly in our hands. But as we step into this new genomic era, what exactly should CRISPR do, and critically, whose hands get to make that decision? Thanks for exploring the blueprint of life with me.